Welcome back to Biocode. In this tutorial, I will continue how to design primers for PCR reaction. In today's video, we are going to talk about circRNA and what software would you use to design a primer for it as a target. First, I need to talk about circRNA itself. What is circRNA? circRNA is a single-stranded RNA that forms a covalently closed continuous loop. This structure is created when the 3' and 5' ends of the RNA molecule are joined together, making it resistant to exonuclease-mediated degradation. circRNAs are generated from precursor messenger RNA, pre-messenger RNAs, through a process known as backsplicing. What's the function of circRNAs? Number 1. Regulation of gene expression. circRNAs can act as microRNA sponges, binding and sequestering microRNAs to regulate gene expression. Protein translation. Some circRNAs have been shown to contain open reading frames ORFs and can be translated into proteins. Biomarkers. Due to their stability and tissue-specific expression, circRNAs are being studied as potential biomarkers for various diseases including cancer. What is backsplicing? Backsplicing is a non-canonical splicing because canonical splicing is for normal messenger RNA. So backsplicing is non-canonical splicing mechanism that generates circRNAs. Unlike the conventional splicing process that joins exons in a linear sequence forming messenger RNA, normal messenger RNA, backsplicing involves joining a downstream splice donor site to an upstream splice acceptor site. The 5' end of an exon is spliced to the 3' end of an upstream exon, forming a loop and resulting in the exon being circularized. As you can see here, this is the normal precursor messenger RNA, consists of exons and introns. Then splicing will happen. In the formation of circRNA, we have different possibilities that lead at the end to form different forms of circRNA. You can see here, mature messenger RNA, it will go through self-splicing. Self-splicing will lead to a backsplicing between exon 1 and exon 2, forming a circRNA consists of exon 1 and exon 2. In another form, intron-bearing driven circularization, you can see here that there are two introns linked together through ALU repeats, and at the end it will form a circRNA. Another form, self-splicing by formation of a lariat. Lariat is like a rope. You can see here this intron is forming a lariat. Okay? A lariat will lead at the end to form an intron circular RNA, not exon circular RNA. The fourth form is exon skipping. And you can see here, exon 2 and exon 3 form backsplicing, and the normal messenger RNA formed between exon 1 and exon 4, which led to the formation of circRNA from exon 2 and exon 3. This was a short introduction about circRNA, but what this video about is designing a primer for circRNA. An important note, the general guidelines for primer design is in the previous video up there. The difference here when we talk about circRNA is that our target is not linear and not resulted from normal splicing. A great tool is used to design PCR primers for circRNA, which is circPrimer 2.0, and here is its citation. You can use this tool either to design new primers or to check the specificity of existing primers, and we will do both of them. So let's open the program. As you can see, the interface of the program is simple. Here The program gives you the ability to work with Homo sapiens or mus musculus, and you can select reference gene from here. I will select the more recent one. Here you have a choice between circRNA as a single target or multiple circRNAs. In this part, you can do primer blasting by checking primer, put your forward and reverse primer here and check the specificity of your primers, and you will get the results here, and here is the annotated results. The third part of the software is when getting you the whole sequence of the circRNA. So let's start by designing a primer for target. Our target will be ARID1A. We are looking to design a circRNA primer for ARID1A. We will start by search. You will get a group of results. All of these results for Homo sapiens, and the last three is for mus musculus. We can choose the first one, and it will get the sequence here in the results port. In this box you can see the annotation of our result. Here our target gene ARID1A. Here is the accession number of the messenger RNA that forms the circRNA. You can change the view here. You are on the default view. You can change the view to function. You can see the coding sequence. It's all of the target is a coding sequence. You can change it again to ORFs. ORFs will show you where the open reading frames are in your circRNA sequence, as you can see here. We will leave it as a divergent primer, as to amplify circRNA you need a divergent primer. We will click template. You can take the template and open primer 3 program here. Paste your sequence here and press pick primers. You will see the results as a divergent primer. Each primer is on a different strand. We have multiple amplicons. If you are using the software to design a primer for a real-time PCR, I prefer the smaller amplicon here, product size 162 base pairs. You can check the melting temperature is okay. I will take this one. This is about support of designing a new primer pair. What about checking the specificity of already existing primer pairs? If you are getting them from a paper, we are going back to the software, but this time we are going to check primer. I will get the primers. Let's open a new terminal. And here our primers. Copy and paste results. And just check. This process will take some time. Oh, and finally we'll get our results. So here you have lenses of the products, as we are checking for primers for real-time PCR. So I will take the first one, the shortest one, 106 base pairs. Click on the target, and here is our results. This is the default view. You can see here that the primer is from exon 2 to exon 4, And if we change to the function tab, you can see it's also all a coding sequence, and I forget to say that it's already our target gene. If we change to ORFs, you can say it's a bunch of ORFs here, and our primers are covering an ORF area. I would like to say that M6A is a position for methylation, by the way. And now you are pretty sure that your primer pairs are good. You can design it and do your experiment safely. I think we covered the program well. To see you in the next tutorial. Thanks.